Salmonella is a gram-negative intracellular organism, and it causes food contamination, food poisoning, and food recall all over the world. And it causes diarrhea and death in both humans, domestic and wild animal. And they do this actually by invading the host intestinal epithelial cells using a number of genes. And there are over 10,000 and 2,000 pangenes and core genes that have been reported. And the role of free-ranging captive wildlife species in these symptomatic carriers of Salmonella remains relatively unrecognized. Now, the objective of the study is to determine the presence of Salmonella in A and in H genes, their virulence genes, and to detect B-lactam resistant genes, Blactem and Black Oxa, and to identify other virulence and resistant genes through whole genome sequencing. 12 Salmonella isolates were used for the study. Salmonella Giogo, Reed, and others as shown. Polymerase chain reaction was used to detect Salmonella virulence and resistant genes, while whole genome sequencing was also used to determine the other genes present as virulence and resistant genes. And analysis was done using SPSs. Now, the results of the study for imaging, which has an expected amplitude of 284 base pairs, we have 10 out of the 12 isolates that showed the band that were positive. For in H, we had 2 out of the 12 that showed the band, and the expected amplitude is 500 BP. For Black, 10. We had the whole of them, the 12 of the isolate, that showed the band and they were positive and the expected band there is 950 BP. And for Black, we had 2 also of the isolates that showed that were positive. Expected band 700 BP. Now, from the whole genome sequencing, we had this number of virulence genes that were detected. For the S-calam, 5 of them, 85, and the other ones, as shown, for the in silico, for the whole genome sequencing, antibiotic resistant genes, we had a total of 2,166 genes identified. And the highest number is the beta-lactams, where we had 1,601 resistant genes identified. Now, conclusion. 10 out of 12 isolates harbored in A, while only 2 were positive for FMH. All the isolates were positive for Black, while 2 were positive for Black Oxa. A total of 119 virulence genes were identified among the isolates, but these include, these as shown, Stahis, Step B, and all these, they code for different resistance. And then we also, that's virulent genes now. These are virulent genes, 119 virulent genes. And all these, they actually enhance virulence. And then we had 2,166 resistant genes coding for different resistance in this bacteria. And we have some of them listed here. We want to recommend that there should be further periodic studies in the molecular epidemiology of Salmonella, especially in wildlife. And there's a need for the timely availability of research data and management of wildlife settings. She also have a working relationship with standard laboratories, and government and non-government organizations should increase funding and funding of research, especially those related to zoonosis and antimicrobial resistance. Thank you.
Thank you very much.